Honors Biology Final Review. You can follow along with this review using your review packet, and you can access the review packet, this video, and notes at srhsbio.wikispaces.com. First section is about ecology. First question is about methylmercury biomagnification. It asks you to create a food chain, labeling the food chain with the producer, primary consumer, secondary consumer, and tertiary consumer. So at the bottom of our food chain is our producers. In this particular example, these are going to be seagrasses and algae. So these are photosynthetic organisms that use sunlight to make food. The arrow goes in the direction of the energy flow. So the primary consumers eat the primary producers. And the animal that's going to represent this, or the organism that's going to represent this, is the periphytons. Periphytons are consumed by crayfish, and crayfish are consumed by our tertiary consumers, which is going to be a bass in this example. Now, based on the food chain, explain how methylmercury can build up to toxic levels. So here we have our primary consumer, and this primary consumer already has 1 unit of food energy. And on the first day, this primary consumer is going to go around and it's going to eat 10 units of food energy. At the end of each day, we lose 90% of that food energy as heat to the environment. So we ate 10 units of food. We're going to see that 9 of them disappear. 1 remains. This remaining unit of food energy can be used by the organism to grow or to meet its other needs in the environment. On the next day, the producer has become contaminated with methylmercury, so that was represented by our color change. So now when the primary consumer uptakes energy, whenever it eats, it's going to eat 10 units of food energy, but it's also going to eat 1 unit of methylmercury, represented by that dark circle. Again, we're going to lose 90% of our food energy, but the methylmercury will be left behind because it cannot be used for energy by the primary consumer. So we see again that 9 of those food units disappear. Okay, next day, again, we uptake energy, 10 food units and 1 methylmercury unit. And again, we lose 90% of the food units only. 1 more day of feeding, we eat 10 units of energy plus 1 unit of methylmercury. We lose 90% of our food energy. So at the end of this 4th day of feeding, my primary consumer has 5 units of food energy and 3 units of methylmercury. So as it feeds, it consumes both food and methylmercury. Each day, the primary consumer loses 90% of the energy from the food. Leftover energy is used to help this organism grow and live. Food is lost each day, but the methylmercury is not used for energy, so it doesn't get digested. Instead, it remains behind. It gets deposited into the tissues of the primary consumer. As we move up trophic levels, we see that this process is magnified. So here is my day 4 consumer. It has 5 units of food and 3 units of mercury. Now, my secondary consumer is going to eat 4 primary consumers. So that's why we've got 4 primary consumers here, because we're going to eat 4 of them for my secondary consumer. So if we eat 4 primary consumers, and each one has 3 mercury units, we end up with 12 mercury units. Same thing for the food. We've got 5 food units per primary consumer, and there's 4 of them that are being consumed, so that's 20. So that means that when the secondary consumer eats the primary consumer, each day it's getting 12 mercury units and 20 food units. So, again, we lose 90% of our food unit energy. So this has no effect on the mercury units because those aren't digested or used for energy. Instead, they are deposited into the tissues. So if we lose 90% of 20, we end up with a loss of 18 food units. So each day, after losing the food units as heat, the secondary consumer has uptaken 12 mercury units and only 2 food units. If we continue this process for 4 days, so we're going to eat the primary consumer for 4 days, we end up with 12 times 4, which is 48 mercury units, and 2 times 4, which is 8 food units, and we started with 10, so that's where we got that 18 from. The secondary consumer had 10 to begin with and gained 8 over the 4 days of feeding. So at the end of the 4th day, my secondary consumer has 18 food units and 48 mercury units. We're going to repeat this process, except this time my secondary consumer is going to be eaten by a tertiary consumer. So one of the day 4 secondary consumers has 48 mercury units and 18 food units. My tertiary consumer is going to eat 2 secondary consumers per day. So that means he would uptake 96 of the mercury units, 2 times 48, and 36 food units, 2 times 18. We're multiplying it by 2, again, because we're eating 2 secondary consumers per day. So that means that each day, the tertiary consumer is going to consume 96 mercury units and 36 food units. Once again, we lose 90% of our food units as heat to the environment. There is no effect on the mercury units. If we lose 90% of 36, that's going to be roughly 32, which means that each day that the tertiary consumer eats the secondary consumers, it obtains 96 mercury units and 4 food units. If we repeat this for a total of 4 days of feeding, we end up with a tertiary consumer that has 384 mercury units and 36 food units. And again, 4 times 4 for 4 days gives me 16, and we started with 20, so that's where we get that 36 from. For 96, we just multiplied that by 4 to get 384. So my tertiary consumer has 36 food units and 384 mercury units at the end of day 4. So as we move up these tropic levels from primary, secondary to tertiary consumer, we see that the amount of methylmercury starts to exponentially increase. So a dietary recommendation for at-risk groups. So first would be to eat at lower tropic levels to get the benefits of seafood. So for our primary consumer, that has the least amount of mercury units compared to the food units. So that would be better to consume a primary or secondary consumer as opposed to this tertiary consumer that's got tons and tons of mercury in it. Avoid eating these tertiary consumers. So these are going to be like tuna, game fish, the large predatory fish that are in the ecosystem. Avoid eating those to avoid eating large amounts of methylmercury. Another option might be to eat younger fish. Younger fish have been feeding in the environment for less time, therefore they have not been able to accumulate as much methylmercury. Next question is about a food web. So the question says, what would happen in the ecosystem if the amount of carbon dioxide in the atmosphere was to decrease? So, of course, carbon dioxide, that is what we need for photosynthesis. So if there is less carbon dioxide available, that means less photosynthesis. Less photosynthesis means that there's going to be less biomass at the producer level. So there will be less plants. The plants are not going to be as productive. So there's not going to be as much food for the successive higher levels in the ecosystem. So with less producers, that means that fewer consumers will be supported by the environment. So the next question asks, what would happen to the prairie ecosystem if the amount of sunlight was to decrease? Again, sunlight is needed for photosynthesis. So with less sunlight, we have less photosynthesis. So it means that we'll have fewer plants. There'll be less biomass at the bottom of the pyramid. So fewer producers means fewer consumers. So if I make the base of my pyramid smaller, all the layers on top of that base are going to have to be made smaller too. So photosynthesis, plants require carbon, water, and sunlight to make sugars and oxygen. So here's the formula for that. I have carbon dioxide with water and light, and we're going to produce sugars. So this is glucose, it's a sugar, and we also produce oxygen. So my question for you is, based on the last slide, which of the following species is likely a photosynthetic producer? So in this graph, we have along the y-axis my population size, and then along the x-axis I have the water depth. And you can see that there is a relationship here between the size of the population and how deep we are in the water. So if we look at something like species C, which is right here, species C is pretty much constant. So species C doesn't have much relationship between population size and water depth. If we look at the species D here, we see that we've got the biggest population at a medium water depth, and then it kind of drops off more or less in each direction. So what is it about water depth that relates to photosynthesis? Well, the deeper down we are in the water, the less sunlight can penetrate. So any photosynthetic producers are going to get less sunlight the deeper down they are. So we would expect that for something that is photosynthetic, as we go down in the water column, the population should get smaller too because there's less sunlight available for photosynthesis. So that is going to be species B. That must be photosynthetic because we see the correct relationship there between water depth and the population size. Next question asks about what would happen in our prairie ecosystem if an invasive species of locusts were to appear. So we see in our ecosystem we do have some locusts that are part of this native population. But what this question is asking is if some other species of locust that wasn't native to this ecosystem were to suddenly be introduced. So this would decrease biodiversity. What would happen whenever that locust move into the ecosystem? It would take over the niche of the native locust. And it might not have any predators, and this could lead to some uncontrolled population growth, which means that these locusts, which feed on plants, would be eating up all of the producers in the ecosystem. And again, if that bottom level gets smaller, the levels on top in the pyramid must also get smaller. So it might wipe out plants by eating them, and this could lead to other species dying off because they don't have access to those producers any longer. So this would decrease biodiversity. We would have fewer species than what we started with, and we would see that those producers would just get wiped out by this uncontrolled locust population that would be an invasive species. Next question is about the differences between primary and secondary succession. So succession is what happens whenever an ecosystem is progressing towards that climax community. So that climax community is going to be the community with the most species, the greatest biodiversity. So primary succession is where we begin with bare rock and we develop into that climax community. There's no soil present whenever we start. And this takes thousands of years. So if you look at this picture, we start with bare rock, and then we have the lichens and mosses. These guys attach to the rock, they grow on the rock, and they break it down and start to make soil. Once that soil starts to appear, we get our grasses, and again, those grasses are preparing the ecosystem for the next step where we get small shrubs, we might start to get fungi, and over time, eventually we develop this climax community of this big forest with lots of different species and lots of different diversity. Notice that as species move in, they make conditions that prepare the environment for the next species to move in. So these mosses and lichens, these are called pioneer species because they're the first species to move into the ecosystem and prepare it for the successive generations of plants and animals. Secondary succession is a little quicker because we begin after the previous ecosystem is destroyed. So maybe there is a fire or a flood, and we still have soil that's left behind, and there might even be some plants that are left behind or seeds that could quickly develop and grow into plants. This takes hundreds of years as opposed to thousands of years. So here we have an ecosystem that's been wiped out after a fire, and there's still some plants left behind. There's some soil there. Those plants fill in and grow, and over a shorter period of time, we again end up with this climax community that is filled with many different species with lots of biodiversity. Next question is about the relationships between organisms in an ecosystem. First, we have predation. So predation is where one organism kills and eats another organism for food. And an example of this would be like wolves hunting and capturing and killing and eating an elk. Mutualism, so a mutual relationship is where both organisms benefit. An example of this might be bees and flowers. So as a bee goes from flower to flower, it's collecting nectar, and it takes that back to its beehive, which it uses to make honey. But while the bee is foraging, it's also spreading around pollen from the flowers. So this allows the flowers to reproduce, it helps spread their pollen around, and both organisms are benefiting. In commensalism, this is where one organism benefits while the other one is neither helped nor harmed. So an example would be barnacles living on a whale. Because the whale is moving and the barnacles are attached to it, there's lots of water going by the barnacles, and that water is filled with food particles. So by growing on this whale, these barnacles are able to capture much more food. The barnacles don't do anything to the whale. They don't hurt it or help it. It might look like they might be harming it because they're growing all over the whale, but there's no harm done to the whale. The whale doesn't affect its ability to get food or mates or any of the other things that whales need to do. Parasitism is where one organism benefits, and that's going to be the parasite, while the other is weakened, and that will be the host. So an example of that could be like leeches or ticks attaching to the dog. Those parasites feed off the blood of the dog. This takes away nutrients that should be available to the dog, but instead are being soaked up by the parasites. And they also usually transmit diseases and things of that nature. So definitely a relationship where the dog or the host is being harmed or weakened by the parasite. So the next question asks, what essential component is missing from this ecosystem? So down here at the bottom, we've got our trees and grasses. Those are my producers. And then I've got my deer, rabbit, mouse, and crickets. Those guys are going to be consumers. The mouse, since it also eats the cricket, would be a secondary consumer. Same thing. The frog is also a secondary consumer. The snake and the mountain lion, according to this food web, those are secondary consumers, and the hawk would be a tertiary consumer because it eats the frog, a secondary consumer. So what we're missing in this organism, or in this food web, is an organism which can take all of these nutrients in these animals once they're dead and recycle them so that they can be reused by the producers. So we are missing decomposers. So if we add that on, we would draw a line from each animal in this ecosystem to the decomposers, because once these things die, it is the job of the decomposers to break them down. So this helps cycling of nutrients and materials between organisms and their environment. Otherwise, once these organisms died, all of those nutrients would be locked away and they would be inaccessible to other things in the environment. So an example could be bacteria, which helps to restore nitrogen compounds to the soil. So what role do bacteria play in the nitrogen cycle? So nitrogen is essential for life. It's in amino acids, it's in proteins, it's in DNA, genetic material, so it is an important element. Nitrogen is available in the air. The air is 70% roughly nitrogen, but it must be fixed into another form so that it can be used by living organisms. So nitrogen as it exists in the atmosphere is not something that organisms can use and access. So this process is called nitrogen fixation, and about 90% of it is done by bacteria in the soil. So this is essential because without these bacteria, none of that nitrogen that's needed for amino acids and proteins, none of that would be available to any of these organisms. So the bacteria fix the nitrogen, and usually this is taken up by plants, which is then taken up by our primary consumers all the way up the food chain. Scavengers, why are they important? So scavengers feed on dead animals. So basically what this does is it kind of speeds up and aids the recycling of environmental materials and nutrients. So this is the job of bacteria and detrivore decomposers, but the scavengers begin this process, and the decomposers and detrivores then complete it once the scavengers have started it. So it kind of speeds things up. Those scavengers break down that dead, decaying matter, and then the decomposers and detrivores can move in. So common scavengers, we think of vultures and hyenas. So those are things that typically are going to feed upon things that are already dead. They're opportunists, so they're looking for any opportunity to scavenge off of something that is already dead. So next question is about food pyramids and biomass. So again, biomass is just the mass of all of the living things at a particular trophic level. So at the bottom, the base has the greatest biomass, and those are going to be our plants or our consumers. In this particular ecosystem, the plants would then be consumed by small fish, which would be consumed by large fish, which then could be consumed by birds. So each level can only keep 10% of the energy that it gets. The other 90% is lost as heat to the environment. So because less energy is available at each level, less biomass can be supported at each level. So to support the upper levels with enough energy, we must have the bottom of the pyramid as the largest. The next section, section 2, is about protein synthesis. Number 9 asks us to label and explain the diagram. So we'll start with number 1. So number 1 here is mRNA. This is coded from DNA during transcription. So what happens is an enzyme attaches onto this strand of DNA, and as it goes down the DNA, it unwinds and it brings in the correct mRNA bases that match up. This is the first step of protein synthesis. Next, the mRNA is going to exit the nucleus through one of the pores in the nuclear membrane. A ribosome will then attach and read the mRNA during translation. So as the ribosome is reading the mRNA, we have these tRNAs, which are attached to amino acids that come in and match up to the set of 3 codons on the mRNA. As the tRNA comes in, it matches up. The amino acids form a peptide bond, and once this is all done, the mRNA and the ribosome detach, the amino acid chain or the polypeptide chain is complete and the protein is made. The order of the bases in the DNA here, they determine the order of the bases on the mRNA. So that in turn determines the amino acid sequence of the protein. Next question is about insulin. So scientists can take the insulin protein for human diabetes and insert that insulin protein into a different organism. So this is typically done with bacteria, and this is how insulin is made for people who have diabetes. So protein synthesis is nearly the same for all organisms. The insulin gene is removed from a human cell, so enzymes are used to kind of cut the gene out, and then different enzymes are used to paste the gene into the DNA of a bacteria. So when the bacteria goes to make its usual bacteria proteins through transcription and translation, that insulin gene has been stuck in there as well, so we can actually trick the bacteria into making insulin. This is really great because we used to have to get insulin from rabbit pancreas, And now we get it from DNA, and nobody gets upset whenever we kill bacteria as opposed to rabbits. So here we've got molecule 1. So molecule 1 represents a double helix DNA strand. Few reasons I know that this is DNA. Number 1, there's T's. So in mRNA there's U instead of T. It's also double-stranded, which is what DNA is, as opposed to mRNA, which would be single-stranded. So a mutation occurs in the second base pair of molecule 1. So what I'm talking about is right in here, a mutation occurs. So this is a point mutation, where one nucleotide changes to another. So perhaps instead of an A, this becomes a T, or something different. This may lead to the wrong amino acid. So we know that every set of 3 of these codons here, every set of 3 of these guys makes 1 amino acid. So perhaps we code for the wrong amino acid and we change molecule 2. In this particular instance, TAC, whenever we code that into mRNA, that becomes AUG, which is the start codon. So there's only one start codon. A mutation here means that the ribosome doesn't know where to start reading. It doesn't have that start codon that says, okay, ribosome, this is where it counts, start to read the mRNA once it's made. So this could potentially have really huge effects, and this protein may not be made correctly or at all. Section 3 is about genetics. So number 12 asks us to compare the consequences of a mutation in a gamete versus the mutation in a body cell. So an offspring develops from a gamete. Whenever 2 gametes meet, that's how you get an offspring. So if there's any mutations in either of those gametes, that is going to affect the entire organism that develops from that gamete, because every single cell develops from those original 2 gametes. So if something's wrong with one of them, it's going to be carried out for all of the cells that develop. Usually many mutations are involved, many chromosomal mutations, so this will have a dramatic effect on the appearance of the offspring. Whereas in a body cell, only a cell that is produced from the specific cell that mutated would be affected. So more than likely in a multicellular animal, this mutated cell would be killed off before it could develop into a cancerous cell, something that would divide and replicate without any control. And if this was a single-celled organism, the organism would likely just die off. So next question is asking us to explain how 2 tall pea plants are able to produce a short offspring. So this is where we're going to need another one of our trusty Punnett squares. So on this side we do our female alleles, and on the other side we do the male's alleles, and then the cross goes here in the middle. So the short height must be a recessive gene. It's masked by the tall gene. So the idea is that even though the 2 plants appear tall, because tall is dominant, it could be masking a recessive short gene. So the way that this would work, 2 heterozygous parents would each have 1 short gene, even though their appearance is tall. So I'm going to do big T for tall, little t for short. Again, these are both heterozygous. So in this cross here, I would have big T, big T, big T, little t, big T, little t again. So all 3 of these offspring are tall. But then I've got these 2 recessive genes, which would give me 1 short offspring. The next question is about polydactyly, which is a gene that causes extra fingers and toes. This particular gene is dominant over the gene for the normal number of digits. So just because a gene is dominant or recessive, that has nothing to do with how common it is in the population. Just because polydactyly is dominant doesn't mean we would expect for lots of people to have extra fingers and toes. Most people are born with a normal number of digits, which is actually recessive. So predict the chances for a couple of parents having children, whether or not their children will have extra toes if the parents are homozygous recessive. So homozygous means the same. Recessive means that they both have little r's. So they both have the normal number of fingers and toes. So a little r mates or crosses with another little r. So I get a little r, another little r. Again, two little r's, and we can kind of predict what's going to happen here. These are all going to be little r's. So that means that 100% of their children will also be homozygous recessive. In other words, they all have the normal number of toes. So the chances of their children having extra toes is 0% because they are homozygous recessive. Next question asks homozygous dominant. So in this case, homo still means same, but the parents are big R, big R. So this means that both of these parents have, or were born with, extra fingers and toes. So again, we're going to get big R, because we're crossing two big R's. One more big R, and just like before, we're going to see a similar pattern here. The only gene that these parents can pass on is the dominant gene. There is no recessive gene available. So that means that 100% of their offspring are going to have extra fingers and toes. Next question, what if the parents are heterozygous? So hetero, of course, means different. So heterozygous parents are going to be big R, little r. So if both parents are heterozygous, so here's one offspring that is big R. Here's another one that is big R, little r. And here's another one that is big R, little r. So 3 out of 4 are definitely going to have extra digits. And one child would be, 1 out of 4, there's a 25% chance that their child would be born with the normal number of fingers or toes. So the chances of their children having extra toes in this particular example would be 75%. Next question is asking about field mice. So in field mice, brown fur is dominant to tan fur. A tan mouse is mated with a brown mouse, producing 16 brown and 14 tan mice. So the question is, what is most likely going to be the genotype of the parents? So if there's 16 brown mice for 14 tan mice, that means that we have roughly 50-50 brown mice to tan mice. So with the Punnett square, a lot of times you can kind of just play around and see what you end up with. So let's say that we have big B, little b for both parents. So we would get big B, big B, big B, little b, big B, little b, and little b, little b. So if we look at this, that's 75% brown fur and 25% tan fur. So this doesn't quite make sense. The question also gives you a clue because it says a tan mouse is mated with a brown mouse. So the only way we can get a tan mouse would be if it was little b, little b. So now I'm going to change one of the parents to little b, little b. So here we would have big B, little b. And this one would still be the same. It would still be big B, little b, except this one would be little b, little b. So here I have 50% tan and 50% brown. So the parents must be, one of them must be homozygous recessive, whereas the other one must be heterozygous. Question 16 asks us to compare and describe the genetic similarity between clones. So clones have 100% the same DNA, so they have 100% the same genes. So an example of this could be identical twins. So after sperm and egg meet, 2 offspring develop from one zygote. So what that means is that these 2 organisms, these 2 offsprings, are identical, just like a clone. Another example would be an organism that can reproduce by mitosis. So for example, if you were to take a violet leaf and put it in water and plant it, water it, take care of it, you can actually grow another violet plant from that leaf. So all the cells of the nearly the newly grown plant would have developed from that original leaf. So they are all going to be identical. Number 17 asks us to figure out the possible genotypes for individual A. So on individual A, we know that one of the parents must be recessive. So I'm going to use little a for this parent. So this is widow's peak, which is a recessive trait. So I know that one of the parents, in this case the female, is little a. Now the male, the father, could be two things. He could be big A, little a, or he could be big A. So if he is big A, that means that all of the offspring would be big A, little a. So this individual could be big A, little a. If the father was, instead of big A, if the father was big A, little a, we'd get a slightly different outcome here. So you might say, okay, well, that individual could be little a. Well, no, he can't, or she can't, because she's not darkened in. So it's not possible that they're little a. So this individual must be big A, little a, regardless as to which one the father is. Whether the father is big A, little a, or big A, the offspring are going to have to be big A, little a. Section 4 is about evolution. So number 18 asks us to compare natural and artificial selection. Describe what roles humans play in artificial selection and compare these to natural processes that take place during natural selection. So in natural selection, the environment has pressure on which organisms are going to be best suited. So the organisms that are best suited to their environment, those are the ones that are going to survive and reproduce. So perhaps these particular organisms are better at blending in, perhaps they can get more food, perhaps they are better at attracting mates. But one way or the other, they're going to survive longer, which means they will reproduce more. So their genes are going to be passed on to more offspring, so that gene will become more common in the population. The fastest natural selection is going to occur whenever there's sexual reproduction, so that means lots of genetic variation in a changing environment. So the changing environment means that the pressure acting upon that population is going to be changing. So as the pressure changes, only those organisms that are best suited to the environment will be able to survive. Next question is about comparing these. So when humans artificially select traits, they are acting as the environmental pressure. They select the desirable traits, and organisms that have undesirable traits are not allowed to reproduce. So this is how many varieties of dogs and crops were artificially selected by humans. So in our little picture here, whenever humans select the traits, it may not be the traits that are best for the environment, it just might be traits that we like. So if we're looking for a small, cute little dog, obviously that probably would not be best suited for nature. Next question asks us about a common ancestry. So first is embryological studies. So if we look at the embryos of different organisms, we see that there are similar patterns of development. So we'll see that eyes develop at around the same time, or limbs develop at around the same time. So this implies that there is some kind of relation and that they have common ancestors, because the basic plan of an organism's beginning remains the same for many different species. So here we have a fish, a reptile, a bird, and a human, and they all have gill slits. Now that doesn't mean that humans have gills, or that at one point we had gills whenever we were in the womb. It just means that in fish, these gill slits eventually develop into gills, whereas in humans, that structure develops into something different. But again, very similar early on in embryological development. So the next question is about DNA similarities. So the more DNA organisms have in common, the more closely related they are. So whenever scientists first went about categorizing different organisms, it was just based on what those organisms looked like, or perhaps how they lived in their environment. Now that we have DNA technology, we're able to actually look at how closely related genetically different species are. So this is the strongest evidence for evolution. So for example, human and chimpanzees share about 98% of the same DNA sequence. So in this picture right here, I have the percent DNA in common for several species. So we see that species A and B have very little DNA in common, so they're probably not very closely related. Species A and E, however, have a high percentage of DNA in common. So A is more closely related to E than it is to any of the other species. Next question is about homologous structures. So these are going to be structures that have the same structure, but they have different functions. So the common ancestry of species can be seen in the structure and the development of these homologous structures, even if their function is different. So if we look at these different organisms here, we have a penguin, alligator, bat, human, cat, whale. So we see that the more closely related the animals are, the more similar their structures. So if you look at the mammals, which are along the bottom, the human, the cat, the whale, and the bat, we see that those structures are much more similar than they are, say, for the penguin. Next one is about amino acid sequences. So DNA codes for proteins. The DNA gives us the amino acid sequence. So because just the way that DNA shows how closely related two organisms are, so does amino acid sequences. So the more closely related two species are, the more similar their proteins, their amino acid sequences, are going to be. Next question is about forming new species. So there's a population of organisms that are permanently split by a natural barrier. The two environments are going to be significantly different. So explain how this could result in two different species. So in my picture here, I've got a population of pine trees, and they become separated by this river that suddenly forms. So on the two different sides of this river, the two species are experiencing different environmental or selection processes. So over time, traits will change to the ones that best match the separate habitats. So perhaps there's something different about the soil quality here, or the amount of sunlight, or something like that. So these two species of pine trees begin to develop. And it's again because they're experiencing those different environmental or selection pressures. Over a very long period of time, these two separate populations of trees may have changed so much they are no longer able to mate with each other. So at that point, they're going to be two separate species. So what is a species? A group of organisms that are similar and they are capable of interbreeding. So obviously if we try to mix a fish with a gorilla, that's not going to work. So only members of the same species are able to interbreed with each other. Next question asks, how do genes, traits, and evolution all connect? So evolution occurs through natural selection. So certain individuals will be selected based on the traits that are best suited to the environment. The individuals with these best suited traits are going to live longer. Maybe they can access food easier, or just otherwise are able to survive better. So traits are due to genetic variations, and if they are favorable, then those particular organisms will be able to survive. So an example would be gray fur, brown fur, white fur. Fur is present on all of the rabbits, but there is variation in the color of the fur, and perhaps those white rabbits are better able to survive the winter because predators can't see them against the light snow. So maybe the white rabbit fur would be a more common trait in the population. This means that these individuals are able to reproduce more. So in other words, they're able to pass on those best suited traits through their genes. So over time, the population will be affected as a whole. So the frequency of certain traits will change depending on which traits best match the environment. So this will lead to evolutionary change. How can exposure to an antibiotic lead to resistant bacteria? So here I have a colony of bacteria, and they're all the same species, but there's some slight variation between them, and that's represented by the different colors. So this colony of bacteria show genetic variation. Now I'm going to treat the bacteria with an antibiotic, and we see that most of the bacteria die off, but a couple of them happen to have some slight resistance due to that genetic variation. Now, that bacteria, the surviving bacteria, repopulates, and now the colony is resistant to the antibiotic. So if I apply the antibiotic, no effect. Now this doesn't just happen in one generation, it's usually due to many generations of bacteria being treated by an antibiotic. So that's why if you have a cold, the antibiotic is not going to do anything, one, because a cold is caused by a virus, but you could also be making resistant bacteria. So that's generally not good. So we try to avoid prescribing antibiotics unless a patient really needs them. And because these bacteria produce asexually, once that resistance is acquired, it is usually kept through successive generations of bacteria. 24 is to define and give an example of an adaptation. So an adaptation is just some trait which helps to increase an organism's fitness to the environment. So it is developed and maintained by natural selection. So an example could be thick fur and a layer of fat to help an organism survive in cold climates, such as this polar bear who really wants you to stop messing with his ice. The next section is about reproduction. So first we have a table to complete, and it's about a couple types of asexual reproduction found specifically in bacteria. So binary fission. This is similar to mitosis. The DNA is duplicated, it's copied, and the cell pinches into two. So in this picture here, we start with one bacteria, we duplicate our DNA, it separates off, we make two bacteria that are identical to the original starting bacteria. So the adult and offspring are identical. There is no genetic variation between generations that reproduce by binary fission. Conjugation is where two bacteria temporarily connect and share genetic information. So this is kind of like sex for bacteria. So what happens is these two bacteria, they form this sex pili, and along that it gives an avenue for them to trade DNA. So now at the end of this trade of DNA, the offspring are going to be different than they originally started as. So the bacteria is now going to develop new traits. So this is how bacteria, even though it generally reproduces asexually, this is how the bacteria is still able to get that variation. They've kind of found this clever way to get around the fact that they only reproduce asexually. So what is the significance of crossing over during meiosis? So meiosis is making sex cells. And the way that I remember this, meiosis, the second letter is E, and in sex, the second letter is E. So this is making gametes or sex cells. So we want as much genetic variation between cells as possible. We want to get the widest range of possibilities for variation in an offspring. That's the whole point of sexual reproduction, is to get that variation. That's why organisms develop, they spend so much energy into making these sex cells and attracting mates and raising offspring, because we need that genetic variation. So this leads to more diversity in a population. So here in this little clip, we can see that these chromosomes have crossed over. Now they're going to split to two different cells. And we're going to do this process twice because this is meiosis. So here we are preparing for the second split. So again, our chromosomes line up, and we're going to split again. So we end up with 4 daughter cells that have half of the required genetic information of a full adult. So whenever 2 of those cells meet, we could remake an offspring or a fully functional cell. So there's crossing over occurring. We trade our arms of our chromosomes. Then we line up in the middle, and we split. So at this point, if we stopped, we would have diploid chromosome number. But we're going to separate again. And what we end up with is 4 haploid daughter cells. So 2 of these daughter cells need to get together to make a diploid cell. Mitosis is a form of reproduction. So mitosis is usually for healing wounds, growing, that sort of thing. But it can also be used as reproduction for some organisms. So an amoeba undergoes mitosis to create 2 offspring. Comment on the similarities between parent and offspring. So in the picture here, we start with amoeba A, and at the end of the cell division, we have amoeba B and C. So mitosis is just making a carbon copy. It's the exact duplication of a cell. Amoebas use mitosis as a form of asexual reproduction. So because this is asexual reproduction, the parent and the offspring are identical, like clones. Next question is about the steps of mitosis. So, first we have prophase. So this is where a cell prepares to divide. They both start with P. So DNA is replicated, the nuclear membrane dissolves. So that's taking place right now. The animation's kind of fast, but you can see at the very beginning that nuclear membrane dissolves away. DNA is replicated. Metaphase occurs when the chromosomes meet in the middle. All those M's there, meet in the middle, is metaphase. Next up comes anaphase, so that's happening right now. So the chromosome move away from each other to the opposite poles of the cell. And we can see those yellow spindle fibers are the ones grabbing onto those chromosomes and splitting them in half. Telophase and cytokinesis is the last phase. So this is where the cell splits into 2. Telophase means 2. And the cytoplasm is evenly divided. So we see that right at the end where the cell splits into 2. So there it is pinching in the middle, 2 separate cells. If an enzyme were to block chromosome separation, how would mitosis be affected? So basically, whenever we get to anaphase, something is preventing those chromosomes from coming apart. So that cell would be unable to complete anaphase and telophase, and that cell would be unable to divide. Last section is called skills and processes. So give an example of bias in an experiment. So bias is a really big problem in experiments. Scientists are always trying to remove bias to make their experiment as objective as possible. So bias is whenever you lean towards something or you do something that alters the outcome of an experiment to meet your assumptions or to meet your needs. So an example would be telling the patient in a drug study that he should expect hair growth. So he might start seeing hair growth even though there really isn't any going on, because now he expects that. That's his assumption from the experiment. Another example could be only publishing the results that meet your needs. So a drug company might hide all of the studies that show negative side effects from a drug and might only publish the ones that show the drug working. A placebo or sugar pill can help to eliminate bias. So if it's a drug study, you give the patient a placebo. That way, anything that those patients report, you're able to compare that to the things that the patients who did take the actual drug reported, and you can eliminate bias through that means. Next question is about the size of a cell. So a child is 1 meter tall, making him 10,000 times taller than a typical animal cell. So what is the size of an animal cell in meters? So basically, a child is 10,000 times bigger than an animal cell. So, if we count the zeros, we've got 4 zeros. So if I were to write that in scientific notation, it would be 1 times 10 to the negative 4. The reason it is negative is because the cell is smaller than the child. So if I asked how many times bigger is the child than the cell, it would be 10 to the positive 4. But because the cell is smaller, we get negative 4.